Welcome to the tutorial on how to complete the transcription and translation activity. It's on the yellow worksheet that you see on your screen. The first step is to remember that there are base pair rules for messenger RNA. If there's a guanine in the DNA, then the messenger RNA is going to have cytosine. And the reverse is also true. If there's a cytosine in the DNA, then there will be guanine in the messenger RNA. One of the other rules is that if there's a thymine in the DNA, then the messenger RNA will code for an adenine. This is where things get different. If there's an adenine in the DNA, the messenger RNA is going to have uracil in place of thymine. So as you look at the sequence of these letters here in the DNA sequence, you're basically going to transcribe the opposite letters, the matching complementary letters in the mRNA sequence in here. So I've done the first couple for you. A guanine is going to respond with a cytosine. A guanine, a cytosine. A cytosine is going to respond with a guanine in the messenger RNA, and so on. So complete this portion before you actually move on to the next step. There are some things you need to know about transcription. First of all, transcription happens in the nucleus. The genes in the template on the DNA are what's used to create the template for the messenger RNA. Once the messenger RNA strand is composed, then we add a 5' cap attached to the front end of the messenger RNA, and the poly A tail is attached to the terminal end. Introns are being removed and recycled in the nucleus, and exons, which are the coding sequences, are spliced together and eventually going to be used exiting the nucleus and expressed to make proteins. Now that you have your messenger RNA sequence in the line up above here, then you will start with the tRNA anticodons. And you're going to remember that you need to look for the start codon in the sequence at the in the messenger RNA. So we found the start codon in the third set of 3, in the third triplet. And we're going to put it in here. This anticodon is just what matches up. Remember, the anticodon is attached to the bottom of the transfer RNA, and at the top of the transfer RNA you have the amino acid methionine. Don't forget, if there's a guanine in the messenger RNA, the anticodon will match up with the cytosine, and the reverse is true. If there's a uracil in the messenger RNA, then the anticodon will have an adenine, and the reverse is also true. Okay, and here are some more facts about protein synthesis, in this case, the translation facts. It happens on the ribosome. And remember that the ribosomes are either free-floating in the cytoplasm or they're attached to the endoplasmic reticulum. The 5' cap with the messenger RNA strand attached enters the small unit, the smaller of the units of the ribosomal complex. The start codon, which is AUG on the messenger RNA, signals the process to begin. Transfer RNA molecules with the anticodon of UAC matches up, which brings the amino acid methionine to the larger unit of the complex. And as the messenger RNA feeds through the ribosomal complex, tRNA molecules will continue to bring amino acids, which match up with the anticodon triplets in the messenger RNA. The transfer RNA molecules will continue to bring the amino acids, which match up using the anticodon at the bottom of the tRNA and matching up with the triplets in the messenger RNA strand. The ribosome uses ATP to bond the amino acids using a peptide bond. So we're going to use the message that's in the messenger RNA, and we're going to use those to read in the codon chart for the amino acids. So when we see the AUG, we're going to go here. We're going to start with the A in this first column. We're going to go to the U in the second position, and then we're going to find the G in the third position. That leads us to AUG, which is the amino acid called methionine. And we're going to place that methionine in the beginning. That's the start codon. We're going to place it in the beginning of our amino acid strand. Once we're done with that, we're going to go to the next triplet, CGU. We're going to say CGU right here, which is arginine. So we're going to write ARG in this ball, in this circle. Then we'll go to CCU, CCU, which is right here, and that's proline. And so we're going to put the PRO right here. And you'll continue along this pathway until you reach an end signal, a stop signal, which is UAA or UAG or UGA. These three codons are stop signals, and when the messenger RNA comes to, when the ribosome reads one of these three triplets, there's no transfer RNA that's brought in. It acts like a set of scissors and cuts the protein off.